At this point, we've looked at atoms. We've used atoms to build big biological macromolecules. Now we're going to take all of those components and we start to build cells. And this is an interesting topic because now we're actually looking at biology. Because biology is life and life is composed of cells. So what we're going to look at, we're going to look at the cell theory, already been discussed a little bit. We're going to discuss why cells are so small. We're going to focus on the two types of cells, prokaryotic and eukaryotic cells. Then we'll take a quick glance into viruses and prions. These are interesting biological entities that cause diseases. So it should be a fun little lecture. What we're looking at here is just a prototypical cell. What you see, these are paramecium found in pond water. You can scoop up some pond water and identify some paramecium. As a prototypical cell, they have a plasma membrane. This plasma membrane surrounds all cells. It separates the external environment from the internal environment. This is identified in every cell you'll see. It defines a cell. Also, since this is a eukaryotic cell, which we'll explore a little bit later, it has a nucleus. You can see that defined nucleus in the center of the paramecium. So we'll continue to look at the parts of cells in the future of this chat here. As we look at cells, we remember the ideas of the cell theory proposed by Schleiden and Schwann. And the cell theory basically states that all living things are made up of cells. Every living thing that we've looked at, from paramecium to humans to plants to fungi, these are all composed of cells. Some are single cells, some are many cells, but cells are these basic units of life. And probably the most important and the dogma-changing idea was cells are derived from pre-existing cells. What this really means is that all cells came from a once existing cell, and it paints this evolutionary picture where there was original cells and every other living organism came from those original cells. So you can see how this kind of changed people's thought historically. So we see this cell. This cell is able to divide to form other cells. This is cell division. We'll take a closer look at this in a couple weeks. Cells are microscopic. You look at your skin and it seems or appears as one sheet of skin. If you were able to look closer and closer, which you can with a microscope, you'll actually see that your skin is composed of individual cells. Unlike a paramecium, which is a single-celled organism, humans are made of trillions of microscopic cells of many different types, from brain cells to skin cells, red blood cells. All of these different cell types perform their job, come together, work together to make you function as a living organism. Kind of cool. And it's interesting how they all work together so well. Most cells, being microscopic, cannot be seen with the naked eye. There are some exceptions. Egg cells are an exception. You can see an egg, and that is a single cell. But what we're really going to be looking at are those cells that are microscopic. We're going to take a closer look when we get into the lab. We're going to start to see some cells. We need a microscope to be able to see these cells because they're small. So you might start to wonder or question, why are cells so small? Why are cells microscopic? That's what we're going to explore next. Most cells are microscopic. In humans, we can look at it. We have many cells. This allows them to perform different roles. But when we look at that paramecium, that was a single-celled organism. All of the functions of that paramecium are performed in that single cell. So there should be no reason why it can't be very large based on this concept. But yet it remains small. So there has to be a different reason why it remains small. The reason that we're going to focus on is this surface area to volume ratio. So the surface area is that plasma membrane, the surface of a spherical cell. It's important for carrying out functions. Primarily, this is where exchange of nutrients and waste occurs. All the nutrients a cell needs to acquire and all the waste it needs to get rid of has to cross that plasma membrane or cross the surface. So we can look at that surface area. This is where everything needs to move past. The volume, the inside of the cell, maybe the cytoplasm, is where the nutrients need to get to and is where the waste is accumulated. So it would be important for a cell to maximize the surface area, have as much surface area as possible to be able to exchange waste and nutrients for as small a volume as possible. Where we're going is we're going to look at why cells are important based on this surface area to volume ratio. So what happens to surface area relative to cell volume as it gets larger? Volume increases faster than the surface area. So as a cell grows, it'll have more volume compared to the growth of that surface area. If we think these 4 blue circles represent 4 cells of equal total volume to that one larger cell, you should be able to see that the 4 cells have much more surface area than that one large cell, even though they're the same volume. We can look another way. Take a big block of ice, set it outside in the summer, and you can see how long it takes to melt. You can take that same block of ice, crush it up into little pieces, put it outside, see which one melts quicker. The reason this can be used as an analogy for surface area to volume is that ice melts at the surface. So the more surface area the ice has, the quicker it's going to melt. Smaller cubes have more surface area. Even if we look at those 2 chunks of ice as having the same volume, therefore the smaller cubes will melt faster, and that broken up chunk of ice will melt faster than the whole chunk of ice. This is why your refrigerators have cubed ice or crushed ice. The crushed ice melts faster, cooling your drink more quickly. The larger ice would last longer. So you can see this extra surface area that these smaller cubes have. So we come back to this question, what happens to surface area relative to cell volume as a cell gets larger? It would not be able to acquire enough nutrients to support that larger volume. It would not also be able to get rid of waste as efficiently. Cells remain small to maximize that surface area. It comes back to that first point. So we can use the equations for surface area to volume of a sphere and compare maybe a cell with a 1 centimeter radius to a cell with a 10 centimeter radius. The radius increased 10 times, but looking at that surface area to volume, that ratio decreased tenfold. So we can see that smaller cell is going to have a larger surface area relative to its volume. So now that we've explored the surface area to volume ratio and why cells remain small, let's look at some different cell types. Prokaryotic cells versus eukaryotic cells. We're going to first look at similarities. All cells have a cell membrane. This surrounds them. So both prokaryotic and eukaryotic cells have a cell membrane. Both of them contain that blueprint. They both contain DNA. Both cell types have ribosomes. Ribosomes are important. They are used to manufacture those proteins. We've already discussed proteins do all of these jobs inside of cells, so therefore you need ribosomes to build them. So both cell types have ribosomes. So these are 3 similarities that we can look at between prokaryotic and eukaryotic cells. Let's look at some differences. DNA is not found in a nucleus in a prokaryotic cell. The term prokaryote means before nucleus. A prokaryotic cell evolved before there was a nucleus, and all a nucleus is a membrane surrounding the DNA. The term eukaryotic means true nucleus. These actually have DNA enclosed in a membrane. So eukaryotic cells evolved after the prokaryotic cells and have increased complexity. I like to use an analogy to compare eukaryotic and prokaryotic cells. When I think of a eukaryotic cell, I think of a house or a mansion. When I think of a prokaryotic cell, I can think of it as like a little hut. Eukaryotic cells, being like a house, have these external walls. You go inside and it's compartmentalized into rooms. A hut, on the other hand, will perform some of the same jobs. You still need to sleep, you still need to eat, but in a prokaryotic cell, it's all performed in one room. That's how a hut would basically work. These little compartments are called organelles. Prokaryotic cells, like a hut, have no internal compartments. Eukaryotic cells have organelles. They have separate rooms for eating, sleeping, as our analogy kind of suggests. Similarly, huts are much smaller than houses. Prokaryotic cells are much smaller than eukaryotic cells. Take a look at a couple examples. Bacteria and archaea are typical prokaryotic cells. These are made up of prokaryotic cells. Organisms that are made up of eukaryotic cells are pretty much everything we're more familiar with. Plants, animals, fungi, protozoans. Protozoans are like those amoeba that we looked at, or paramecium that we looked at earlier. So as we look at a typical bacterial cell, we notice that it has DNA, ribosome, plasma membrane. It has some other structures that are important to the role a bacteria plays, but we do want to see those structures that are found in all living cells. A typical animal cell, it's going to be much larger. It has those internal compartments, or as we call them, organelles, where they're going to have specific jobs. Each organelle is like a room in the house, has a specific job. We're going to discuss those roles in greater detail next week. For now, just kind of understand these similarities and differences. The last thing we're going to look at are some interesting biological entities, things that are not considered living organisms, but yet are important to us. Viruses, something you've probably heard of in the past, very interesting. They're not considered living organisms, primarily because they're not composed of cells. Basically, they're DNA or RNA, so some type of nucleic acid surrounded by a protein coat. Some of them have a little bit more to them than that, but simply that's what they're primarily made of. They also don't have any function. They're dormant outside of our host cells. So they depend entirely on other organisms for replication, for raw materials, for everything needed to make more viruses. So that's why we get sick from viruses, because they're sabotaging our cells, taking them over and using our material. There are many diseases caused by viruses, AIDS caused by viruses. You may be familiar with influenza, the common cold. These are caused by viruses. So viruses need that host cell. They have to attach. They have to gain entry, releasing their DNA. And then that DNA is treated like it's our own DNA, and our cells actually manufacture new viruses. So kind of cool. The last thing to look at is protein diseases. Wow, even proteins can cause disease? That's crazy, right? So proteins are able to cause disease. Bovine spongiform encephalopathy. You may have heard of it as mad cow disease. In humans, a similar type of disease is called Creutzfeldt-Jacobs disease. These are caused by infectious proteins. That's pretty interesting. The proteins are called prions, and these prions are actually just abnormally shaped proteins, normal proteins that have an abnormal shape. These propagate or replicate by changing the shape of normal human prion proteins. When the shape is changed, they aggregate and they start to cause damage in the brain. So we can see this normal shaped protein and the abnormally shaped prion protein. So kind of interesting ideas. We'll look at the viruses and prions a little further in videos. Just a little introduction into those diseases. Hopefully this was interesting and a good discussion, and look forward to talking to you next week.